friends, I am Tehseen Khan, Assistant Professor. Today, we are going to deal with an interesting and learning episode of B.Pharm on the important title Phytochemical Screening 2, scripted by Dr. G.P. Chaudhary and Ashutosh Paljain. So, let's start our episode while taking a look at what we are going to learn today. We will begin with the general introduction of the topic, followed by general principles of formation of primary and secondary plant metabolites and biosynthetic pathways of secondary plant constituents. Now, let's start with the introduction of the topic Phytochemical Screening 2. Introduction. Plants produce a vast and diverse assortment of organic compounds, the great majority of which do not appear to participate directly in growth and development. All organisms need to transform and interconvert a vast number of organic compounds to enable them to live, grow and reproduce. They need to provide themselves with energy in the form of ATP and a supply of building blocks to construct their own tissues. An integrated network of enzyme-mediated and carefully regulated chemical reactions is used for this purpose, collectively referred to as intermediary metabolism, and the pathways involved are termed metabolic pathways. Some of the important molecules of life are carbohydrates, proteins, fats and nucleic acid. The word biosynthesis comes from the two root words bio, which indicates that the reaction is taking place within a living organism, whereas synthesis, which indicates the simple starting materials and being combined to form larger products. A biosynthesis pathway is the description of the steps of the chemical reactions that occurs when a living organism creates a new complex molecule out of simpler, smaller precursors. These molecules are synthesized in the entire living organism through the biosynthetic pathway. Plants are very efficient at synthesizing organic compounds via photosynthesis from inorganic materials found in the environment. Primary plant metabolites are simple molecules or polymers of simple molecules synthesized by plants. Generally, do not have therapeutic activity but essential for the life of plants. These processes demonstrate the fundamental unity of all living matter and are collectively described as primary metabolism, and the compounds involved in the pathways being termed primary metabolites. Thus, degradation of carbohydrates and sugars generally proceeds via the well-characterized pathways known as glycolysis and the Krebs or citric acid or tricarboxylic acid cycle, which release energy from the organic compounds by oxidative reactions. Oxidation of fatty acids from fats by the sequence called beta oxidation and also provides energy. Aerobic organisms are able to optimize these processes by adding on further process oxidative phosphorylation. Metabolic pathways are thus available to interconvert amino acids or degrade those not required and thus provide a further source of energy. Most organisms can synthesize only a proportion of the amino acids, which is a basic requirement for protein synthesis. Secondary metabolites are complex organic molecules biosynthesized from primary plant metabolites in plant cells. Secondary metabolites are classified as alkaloids, glycosides, tannins, phenolic compounds, volatile oils, terpenoids, saponins, steroids, resins and bitter principles, which synthesize, degrade and generally interconvert compounds commonly encountered in all organisms. They also exist an area of metabolism concerned with compounds which have a much more limited distribution in nature. Such compounds called secondary metabolites are found in only specific organisms. The most important building blocks employed in the biosynthesis of secondary metabolites are derived from the intermediates acetyl coenzyme A, shikimic acid, mevalonic acid and 1-deoxyxylulose 5-phosphate. The major source of carbon is usually glucose, which in field of biochemistry have explained enzyme catalysis and their interrelated reactions resulting in formation of primary metabolites and also their role in synthesis of secondary products. The primary and secondary metabolites form carbon metabolism in plants. Here, the sugar forms phosphophenol pyruvate by glycolysis and then pyruvate and TCA cycle. Here, the sugar forms polysaccharide nucleic acid and pentose phosphate, where polysaccharide nucleic acid forms glycosides sugar derivatives and pentose phosphate by the process of shikimic acid pathway forms tannins, vanillin, lignin and flavonoids and also aromatic amino acids and proteins. Here, the pyruvate intermediate forms aliphatic amino acids. Now, the TCA cycles forms malonyl coenzyme A, which forms fatty acids and then lipids. And here, acetyl coenzyme A forms mevalonic acid and steroid isoprenoids. Understandings of phytochemical screening 2. Following aspects are highlighted in this lesson. First, shikimic acid pathway. Second, acetate mevalonate pathway. Third, biosynthesis of glycosides. Fourth, biosynthesis of alkaloids. Shikimic acid pathway. Shikimic acid is produced from a combination of phosphoenol pyruvate, that is PEP, and glycolytic pathway intermediate and erythrose 4-phosphate from the pentose phosphate pathway, in which the phosphate is removed from PEP and methylene carbon, then form a bond with the carbonyl carbon of erythrose 4-phosphate 3-deoxyarabino heptylosonic acid 7-phosphate, that is DAHP, is synthesized in presence of DAHP synthase. The phosphate is removed from C7 of DAHP and the resulting carbocation from a bond with C2. The enzyme then catalyzes the oxidation of the hydroxyl group on C6 to a ketone and a reduction of ketone on C2 to an OH to yield 3-dehydroquinic acid. Reduction of 3-dehydroquinic acid leads to quinic acid, a fairly common natural product found in the free form as esters or in combination with alkaloids such as quinine. A dehydrogenase reductase binds dehydroquinic acid that catalyzes removal of the hydroxyl next to the hydroxyl group on C1 and a proton from adjacent C6 carbon, generating a double bond in a ring. The NADPH dependent reductase activity then reduces the keto group of a hydroxyl group to generate shikimic acid. Shikimic acid itself is formed from 3-dehydroquinic acid via 3-dehydroxyshikimic acid by dehydration and reduction steps. The simple phenolic acids, protocaticoic acid, that is 3,4-dihydroxybenzoic acid, and gallic acid, that is 3,4,5-dihydroxybenzoic acid, can be formed by branch point reactions. 3-dehydroshikimic acid, which involve dehydration and enolization. In the case of gallic acid, gallic acid features as a component of many tannin materials. The reactions of the pentose phosphate cycle may be employed for the degradation of glucose, but they also feature in the synthesis of sugars by photosynthesis. Shikimic acid is further metabolized to corysmic acid, another key intermediate in this pathway. Phenylalanine, tyrosine and tryptophan are 3 aromatic amino acids synthesized from the shikimate pathway, which is only found in microorganisms and plants. The shikimate pathway is not only a source of these amino acids, also of many phenolic compounds, including pigments, defensive compounds, phenols, cinnamic acid derivatives and lignans alkaloids. Under normal growth conditions, similar to 20% of the carbon fixed by plants flow through this pathway. Schematic diagram of a phenyl propanoid or benzenoid biosynthetic pathway in plants. Phenylalanine, which is synthesized in the plastid through the shikimate and aromatic amino acid biosynthetic pathways, is the precursor for both volatile right hand side, and non-volatile, which is on left hand side, specialized metabolites. The chemical structure of representative volatile compounds that contribute to the aroma and flavor of flowers and fruits is shown. Dashed arrows represent several enzymatic steps. Acetate mevalonate pathway. The terpenoids from a large and structurally diverse family of natural products derived from 5-carbon isoprene units joined in head to tail fashion. They are classified into following classes on the number of isoprene units. C5. First is hemiterpenes C5, monoterpenes C10, sesquiterpenes C15, diterpenes C20, triterpenes C30, tetraterpenes C40. 3 molecules of acetyl coenzyme A are used to form mevalonic acid, that is MVA. MVA had been established as precursor of the sterol cholesterol. 2 molecules of acetyl coenzyme A undergo Claisen type of condensation to give rise to acetoacetyl coenzyme A, to which a third molecule is incorporated via a stereospecific aldol addition, giving the branched chain ester beta hydroxy beta methyl glutaryl coenzyme A, HMG coenzyme A. This upon 2-step reduction will give rise MVA, the 6-carbon compound. MVA is transformed into the 5-carbon phosphorylated isoprene units in a series of reactions. Beginning with phosphorylation of primary alcohol group, 2 different ATP dependent enzymes are involved, resulting in MVA diphosphate and decarboxylation or dehydration, then follow to give isopentenyl pyrophosphate, that is IPP, which upon isomerization will form dimethyl allyl pyrophosphate, that is DMAPP. Biosynthesis of GPP or NPP, where dimethyl allyl pyrophosphate, that is DMAPP, and isopentenyl pyrophosphate, that is IPP, undergoes reaction to form geranyl pyrophosphate, that is GPP. Biosynthesis of various type of terpenoids. Geranyl pyrophosphate, that is GPP, also known as geranyl diphosphate, GDP, is an intermediate in the HMG coenzyme reductase pathway used by organisms in the biosynthesis of farnesyl pyrophosphate, geranyl pyrophosphate, cholesterol, terpenes and terpenoids. Thus, mono, sesqui, di, tri and tetraterpenes are biosynthesized using the following substrates. This is achieved by an enzyme known as terpene clycase. A large family of enzymes that use GPP, FPP and GGPP as substrate for the formation of mono, sesqui, diterpenoid products. The enzyme terpene cyclases use magnesium ion or manganese ion as cofactor during catalysis. Here you can see the formation of monoterpenes, sesquiterpenes, diterpenes, triterpenes and tetraterpenes. The volatile oils containing principally isoprenoid compounds are shown in figure. Preliminary stages in the biosynthesis of isoprenoid compound. Here, acetate forms acetoacetyl coenzyme A, which further forms beta hydroxy beta methyl glutaryl coenzyme A, which on reduction gives mevalonic acid, and further forms 5-phospho-mevalonic acid, which gives 5-phospho-3-phospho mevalonic acid. Here, 3-isopentenyl pyrophosphate and 3,3-dimethyl allyl pyrophosphate forms geranyl pyrophosphate, which forms monoterpenes, straight chain cyclic and bicyclic, and it again forms phenyryl pyrophosphate, that is C15, which gives sesquiterpenes, open chain, monocyclic and bicyclic. Before moving to our next module about biosynthesis of glycosides, I would like to suggest you to visit our website www.cec.nic.in for further readings and learning of more topics associated with your B.Pharm syllabus. Here you will find many lectures, e-content, FAQs, LORs and many more. Hope this site will be a catalyst of learning for you. Now coming to our next module, that is module 4, which is biosynthesis of glycosides. Glycosides are organic compounds from plants which on enzymatic or acid hydrolysis give one or more sugar moieties along with non-sugar moiety. The former is called as glycone and the later as aglycones or genins. Terms used for aglycones are generally self-explanatory, the names. Saponin means soap-like consistency. Cyanogenetic means producing hydrocyanic acid. Cardiac glycosides having action on the heart. The synthesis of glycosides in plant cells involves interaction of nucleotide glycoside such as UDP, glucose with alcoholic or phenolic group of second compound aglycones. The principal pathway of glycoside formation involves the transfer of uridylyl group from uridine triphosphate, that is UTP, to sugar 1-phosphate, and the enzymes catalyzing this reaction are known as uridylyl transferases. The subsequent reaction regulated by enzymatic system glycosyl transferase involves transfer of sugar from uridine diphosphate to a glycone moiety resulting in formation of glycoside. The principal pathway of glycoside formation, which represents the UTP plus sugar phosphate, which forms UDP sugar plus phosphate, and second one is UDP sugar plus aglycones, which forms glycosides plus UDP. Cyanogenic glycosides. Cyanogenetic glycosides are phenylpropanoid compounds derived from amino acids, phenylalanine and tyrosine, which are the products of shikimic acid pathway. The aglycones of linamarin are derived from valine and lotustyrylene from isoleucine. B. Flavonoid glycosides. Flavonoids are mostly yellow colored substances and are the derivatives of phenol and presents in the form of glycoside. These are synthesized by the phenylpropanoid metabolic pathway, in which the amino acid phenylalanine is used to produce 4-coumaroyl coenzyme A. This can be combined with malonyl coenzyme A to yield the true backbone of flavonoids, a group of compounds called chalcones, which contain 2 phenyl rings. Conjugate rings closure of chalcones results in the familiar form of flavonoids, the 3-ringed structure of a flavone. The metabolic pathway continues through a series of enzymatic modifications to yield flavonones, dihydroflavonols, anthocyanins, enzymes giving chalcone synthase couple a cinnamoyl coenzyme A unit with 3 malonyl coenzyme A units, giving naringenin chalcone, which act as precursor for a wide range of flavonoid derivatives like flavonones, luteolins, asparagine, flavonols, quercetin, camphorol, catechins, alpha-zalcine, anthocyanidins, pelargrodinin. C. Saponins glycosides. Saponins are glycosides which even at low concentration produce a frothing in aqueous solution, have soap-like properties. Saponins are divided into 2 categories based on chemical moiety. These are steroidal saponins and triterpenoids saponins. All steroidal saponins have the same configuration at spirocenter carbon 22, but stereoisomer at carbon 25 exists. Example, yamogenin. The spiroketal function is derived from the cholesterol side chain by series of oxygenation reaction, hydroxylating carbon 16 and one of the terminal methyl and producing a ketone function at carbon 22. Diosgenin is the principal example of a steroidal saponin and is obtaining from Dioscorea plant. D. Cardiac glycosides. The aglycones of cardioactive glycosides are steroidal in nature. They are the derivatives of cyclopenanthrophenine ring containing an unsaturated lactone ring attached to carbon 17 cholesterol production through acetate, mevalonate, isopentenyl, pyrophosphate and squalene. The therapeutic action of cardioactive glycosides depend on the structure of A-glycone and type of sugar units attached. Two types of A-glycone are identified. Cardenolide, example digitoxygenin, that is C23, and bufadienolides, example helibrigenin, that is C24. Biosynthesis of alkaloids. A. Alkaloids derived from ornithine. Ornithine is incorporated stereospecifically and asymmetrically into pyrrolidine ring of tropane nucleus, the alpha carbon of ornithine becoming the carbon 1 of tropane nucleus. Methionine acts as the methyl group donor, whereas phenylalanine is a precursor of tropic acid. In some families, the malonyl coenzyme A units that form compounds such as hygrine condense internally to form a bridged structure. Tropinone is a key intermediate in the biosynthesis of many tropane alkaloids in the Solanaceae. Atropine is a mixture of R and is hyoscyamine. An unsymmetrical putrescine intermediate is incorporated into cocaine in erythroxylum coca. B. Alkaloids derived from tryptophan. L-tryptophan is a neutral heterocyclic amino acids containing essentially an indole ring system. It has been observed that it serves a precursor for a wide spectrum of indole alkaloids. Major rearrangements reaction may convert the predominant indole ring system into a quinoline ring system, thereby enhancing further the overall ability of tryptophan to act broadly as an alkaloid precursor. C. Alkaloids derived from phenylalanine, tyrosine and related amino acids. These amino acids and their corresponding decarboxylation products act as precursors various alkaloids including ephedrine, colchicines and opium alkaloids. D. Alkaloids derived from tryptamine. Here the figure number 16 represents biosynthesis of beta-carboline. Biosynthesis of beta-carboline alkaloids occurs through the formation of Schiff base from tryptamine and aldehyde or keto acid and subsequent intramolecular Manish reaction, where the carbon atom of indole serves as a nucleophile. Then aromaticity is restored via the loss of proton at the C atom. The resulting tetrahydro beta carboline skeleton then gradually oxidizes to dihydro beta carboline and beta carboline. In the formation of simple beta carboline alkaloids such as harmine and harmaline, pyruvic acid acts as the keto acid. Biosynthesis of monoterpenoid indole alkaloids begins with the Manish reaction of tryptamine and secolignin. It yields strictocidine, which is converted to 4,21 dihydrogesosizine. Then the biosynthesis of most alkaloids proceeds through cyclization with the formation of cathinamine and subsequent reduction of agmalicine in the presence of nicotinamide adenyl dinucleotide phosphate, that is NADPH. In the biosynthesis of other alkaloids, 4,21 dihydrogesosizine first converts into preaquamycin, which gives rise to strychnine alkaloids. By bis indole alkaloids vinblastine and vincristine are produced in the reaction involving catarantine and vindoline. Alkaloids derived from tetrahydroisoquinoline. The opium alkaloids morphine, codeine and other opium derived by the coupling of tetrahydroisoquinoline skeleton, followed by reduction. The R-reticulene is established as a precursor of the morphine alkaloids, coupling ortho to the phenol group in the tetrahydroisoquinoline and para to the phenol in the benzyl subsequent then yield salutaridine. This upon reduction will form salutaridinol, which on combination with acetyl coenzyme A form thebine. the oxidation followed by hydroxylation and reduction yield codeine, then demethylation form morphine. Dear friends, we have reached to our last section, that is summary. Summary. In this chapter, we described basic properties of natural product molecules, which are biosynthesized by a sequence of reactions, which are catalyzed by enzymes. We have focused mainly on all type of biosynthesis pathways. Here we studied more details for better understanding to the diversity and complexity of natural product structures. Overview of the origin of compounds starting from the set of intermediate building blocks acetyl coenzyme A, MVA and MEP, shikimic acid and the amino acid phenylalanine and tyrosine, tryptophan, ornithine and lysine. In the remainder of this chapter, we will discuss the major type of plant secondary metabolites and their biosynthesis. With all this information, here we come to the end of today's lecture, Phytochemical Screening 2. I will be back with few more lectures in this series. Do keep in mind what we discussed today. Time for you to self-study. If you want to learn more and enhance your knowledge, you may log on to our website for MCQs, quizzes, LORs on www.cec.nic.in. Till then, keep studying. Goodbye.